Good afternoon, people. I am your news anchor, Ralph Oro, bringing you the latest information for Daily Prime News.
Magandang buhay, people! I am your news anchor, Rosh Hashana. Will give you hottest news of recombinant DNA. Sa ulo ng nagbabagang balita. In the head of a flaming news.
Recombinant DNA is the method of joining two or more DNA molecules to create a hybrid. The technology is made possible by two types of enzymes, restriction endocyclase and ligase. A restriction endocyclase recognizes a specific sequence of DNA and cuts within or close to that sequence.
The discovery of recombinant DNA describes the purpose of its profound form into the data. Recombinant DNA is a form of DNA constructed in a laboratory. It is generated by transferring selected pieces of DNA from one organism to another. By chance, a restriction enzyme's recognition sequence will occur every basis along a random chain. The amount of fragment ends in moles and is generated by cutting DNA with a restriction enzyme. To understand more, Shin will have an expert to explain to us this technology. Shin?
Yes, Rosh. To understand more about recombinant DNA, let us interview a very important person, a famous doctor from BioChem Hospital, Dr. Christine Calarian. Doc Chris, please give us a comprehensive information about the process and factors of recombinant DNA.
Thank you, Sheena. Thanks for also having me here today. So hello everyone, I am Dr. Christine and I will be talking about the process of recreating recombinant DNA. So what is recombinant DNA? In technical definition, it is a DNA molecule formed by laboratory methods of genetic material from multiple sources, creating a sequence that would not otherwise be found in biological organisms. So this definition can be boiled down to taking one piece of DNA and combining it with another strand of DNA. First, we need some vocabulary. So a plasmid is a circular self-replicating form of DNA found in bacteria. So it is important to notice that this is the chromosomal bacteria DNA pictured here in red. There are also enzymes. You might have encountered this term with other biological processes, but here, enzymes act as a catalyst to cut specific DNA sequences. This process of DNA cutting is also called digestion. To help you how enzymes work, you could think of them as a molecular-sized scissors specialized to specific DNA codes. So here, this is an overview of the process, but we will go through it step by step. So first, the targeted gene from the human DNA, in this case, human growth hormone, is isolated. And then a bacterial plasmid is extracted from a bacterial cell. Enzymes are applied next. So the enzymes here is EcoR1. So what is EcoR1? It is a restriction endonuclease enzyme isolated from species E. coli. And then going back, remember, enzymes act as a scissors cutting the DNA at a specific sequence. The cuts from the enzymes create a sticky ends or overhangs in the DNA. The sticky ends of the human gene fit into the sticky end of the plasmid. On the plasmid because they were cut by the same type of enzyme. and then the sticky end of both plasmid and the gene come together, forming a complete plasmid. And this process is called DNA recombination, and the plasmid product is recombinant DNA. So the recombinant DNA plasmid is then inserted into a bacterial cell. The bacterial cell will then clone itself many times over, therefore making many copies of the recombinant DNA plasmid. So again, here is the overview of the steps we have covered. So that was it for the recombinant DNA. I hope you learned something. Thank you.
So that's it pala, Doc. Thank you for the information and for allowing us to interview you today. Thank you, Doc Chris. Now, let us hear about the history of recombinant DNA from a famous scientist known worldwide, Dr. Andrea Enocean.
Hello everybody, I am Andrea and I am going to share the brief history of recombinant DNA. The first production of recombinant DNA molecules using restriction enzymes occurred in the early 1970s. The recombinant DNA technology involves the joining of DNA from different species and subsequently inserting the hybrid DNA into a host cell, often a bacterium. Researchers at UC San Francisco and Stanford use restriction enzymes to cut DNA from different species at specific sites, and then fuse the cut strands from different species back together. Stanley Cohen of Stanford and Herbert Boyer of UCSF applied for a patent on recombinant DNA technology in 1974, and it was then generated in 1980. Boyer would co-found Genentech way back in 1976. So the Cohen-Boyer patents eventually had more than 200 licenses, biotechnology and pharmaceutical companies, and earned Stanford and UCSF more than 100 million in royalties. Paul Berg, a biochemist at Stanford who was among the first to produce a recombinant DNA molecule in 1972. He wrote a letter along with 10 other researchers to the journal Science, and in the letter it stated that they urged the NIH, or the National Institute of Health, to regulate the use of recombinant DNA technology, and meanwhile they urged scientists to halt most recombinant DNA experiments until they better understood whether the technique is safe or not. So these concerns eventually led to the 1975 Asilomar Conference, where 100 scientists gathered to discuss the safety of manipulation DNA from different species. The meeting resulted in a set of NIH guidelines and revised the document titled Guidelines for Research Involving Recombinant DNA Molecules several times already since 1976. So Paul Berg was awarded a Nobel Prize in Chemistry in 1980 for his fundamental studies of the biochemistry of nucleic acids with particular regard to recombinant DNA. So that is all for the brief history of recombinant DNA. I hope you guys learned something!
And we are back again at the station. I am Diane Hernandez and this is Daily Prime News. Ano kaya ang mga kaso o types of diseases nito? Let us have Miss Jasmine Sanoy para ma-explain naman sa atin ang mga types of diseases sa ganitong kaso. Miss Jasmine?
Thank you, Ms. Hernandez. Ngayon, aalamin naman natin kung ano nga bang merong sakit sa recombinant DNA. The pharmaceutical products synthesized through recombinant DNA technology completely changed the human life in such way that the U.S. Food and Drug Administrative, or the FDA, approved more recombinant drugs in year 1997 than in several years combined, which includes anemia, AIDS, cancer, or the Kaposi's sarcoma. The inherited human disease of DNA repair include many cancer susceptibility syndrome, such as the hereditary non-polyposis colon cancer and breast or ovarian cancer. So that are the different types of diseases in your recombinant DNA. Back to you, Ms. Hernandez.
So yun nga people, so how about the possible treatments for this type of disease? Let us call out our spokesperson, Ms. Denerica.
Good day. So these are the treatment for diseases. AIDS. Recombinant Salmonella bacteria can be harnessed to vector HIV antigens or DNA vaccines to the immune system for induction of specific protective immunity. These are capable of activating the innate, humoral, and cellular immune responses at both mucosal and systemic compartments. For anemia, erythropoietin has been produced into vitro. It is the outside of our body using recombinant DNA technology. The purified recombinant hormone has promise for persons with chronic renal failure who develop anemia because of lack of erythropoietin. So in cancer, recombinant immunotoxins are composed of a very potent protein toxin fused to a targeting moiety such as recombinant antibody fragment or growth factor. These molecules bind to surface antigens specific for cancer cells and kill the target cells by catalytic inhibition of protein synthesis. These recombinant products are used with the main objectives of killing the tumor and stimulating immune cells to respond to the cancer cells. So these principal recombinant products in anti-cancer therapy are immunostimulants, vaccines, antibodies, immunotoxins, and fusion proteins.
Nandito po ako sa isang laboratory facility at ilalahad ko ang mga recent discoveries ukol sa recombinant DNA. So, ang recombinant DNA technology has been proven important to the production of vaccines and protein therapies such as human insulin, interferon, and human growth hormone. These certain advantages of recombinant DNA has been used up until today. However, a more recent discovery on March 2014 states promising results announcing from trial conducted with HIV patients. Moreover, advances in recombinant DNA technology, for example, the creation of recombinant artificial chromosomes combined with semi-automated and later automated DNA sequencing. In agriculture, recombinant DNA has increased the overall production of crops as well as decreased the amounts of herbicides and insecticides used by farmers. Sounds amazing, right? Wait, there's more. Recombinant pharmaceuticals are now being used confidently and rapidly attaining commercial approvals using rDNA or recombinant DNA to create human insulin rather than obtain it from animal sources allows the widespread use across the pharmaceutical industry. Recombinant DNA technology is likely to also have profound effects on society, including better health through improved disease diagnosis, much better understanding of human gene variation, improved drug and pharmaceutical production, and vastly more sensitive and specific crime scene forensics. O diba? At yan lamang yan. Ako po si Lorena Norder na nagsasabing ang buhay ay okay-okay ra.
And we are back in Daily Prime News, and we've come to the conclusion for recombinant DNA technology. It has proven an important purpose to the production of vaccines and protein therapies such as human insulin, interferon, and human growth hormone.
It is also used to produce clotting factors for treating hemophilia and in the development of gene therapy. And that's it, people! I am your news anchor, Rosh Hashanah. Magandang araw po!
And I am Ralph Ora, and this is Daily Prime News.